So thank you, Wolfram, Denny, and Michael for inviting me to speak at the 2020 PCTC CVBE Symposium. And I just want to move to advance my slide. So this is my disclosure statement here. And so I think as many of you know, there's a significant interest in precision medicine or precision health. And this concept was first promoted by President Obama back in 2015. And much of this has been made possible by recent advances in next generation sequencing, bioinformatics, big data, and then by understanding our genetics along with lifestyle and environmental changes. The hope is that we can better predict which treatment might work best for all of us. And in the case of the DNA sequencing, the technology has become much cheaper with a lower error rate, broader coverage, and much faster turnaround time. So that by back in 2000, right here, it cost about $1 billion and over 13 years to get your DNA sequence. And then today you can get the DNA sequence for less than $1,000. I would say with increasing usage of next generation sequencing, there's hope that the clinical care could be more predictive, preventive, personalized, and participatory, the 4 Ps of precision medicine. Now, back in 2014, we were fortunate to receive a R24 Shared Resource Grant with the support of Dr. Denny Buxton and NHRBI. Since then, we have generated more than 1,000 IPS lines as open shared resource. We have a standard operating procedure or SOP for generating these lines. We typically use human PBMC as the starting cell source. We use Sendavirus as the reprogramming method. We then differentiate them into cardiomyocytes, cardiac fibroblasts, smooth muscle cells, and endothelial cells. And then we have also standardized assays and protocols to assess cardiomyocyte morphology, calcium handling, metabolism, single cell contractility, 3D contractility in collaboration with the Wolfram, and then arrhythmia. Now, with that as a background, I will briefly highlight how we will use, we have been using iPS cells for disease modeling, followed by cardio oncology, and then the concept of the clinical trial in a dish. Shown here is a schematic of how the different causes of heart diseases, which I would say falls into 3 buckets. The first category is the primary cardiomyopathies, which could be genetic or acquired. Next category is the secondary cardiomyopathies, which could be amyloidosis, hemochromatosis, and drug-induced cardiac toxicity. And then the third category is the more bread and butter causes, such as coronary artery disease, hypertension, and valvular heart disease. Now, in a study done by former postdoc J.Cho Lee, who's now a faculty in Korea, here we recruited a large family cohort with LAMIN AC mutation. Many of these patients have already developed heart failure and arrhythmia, and one of them required heart transplant. And then using iPSC-derived cardiomyocytes, we confirmed that the mutation in LAMIN is indeed responsible for cellular arrhythmia and impaired contractility. To make a long story short, what we found was that the healthy control cardiomyocytes, they have normal expression of lamin protein, normal nuclear structure, and normal lamin-associated domain, as shown in this cartoon here. However, patients with lamin AC mutation have abnormal nuclear structure that cause increased open chromatin in the lamin-associated domains instead. This then leads to activation of genes that should have been silenced in otherwise healthy conditions. And using RNA sequencing and ATAC-seq, we identified PDGF signaling pathway as one of those abnormally activated genes in the mutant cardiomyocytes compared to the control cardiomyocytes. And then importantly, we also show that the blocking the PDGF pathway with FDA-approved drugs such as tyrosine kinase inhibitors, quinolonib, and sunitinib at very low doses, not the doses that we would use for chemotherapy, could rescue the abnormal phenotype of these mutant cardiomyocytes. Now, more recently, 2 other postdocs in the lab, Junri and Dilip Thomas, were able to show that human iPSC cardiomyocytes can be used to model non-genetic cardiomyopathies. In this case, iron overload cardiomyopathy, which is quite common in patients with hereditary hemochromatosis or patients with beta thalassemia who require multiple blood transfusions. In this case here, we found that the iron overload actually causes significantly altered calcium kinetics, which explains the contractile dysfunction and arrhythmia. And then after screening for several drugs, we found that epsilon, which is a selective divalent metal transporter 1 or DNP1 inhibitor, and also an antioxidant, and can be used to effectively prevent the iron overload phenotype and may provide a promising therapy moving forward. I want to switch gear to the second topic, which is using human iPS cells for studying cardio oncology. This is a major problem in the US because heart disease and cancer account for the top 2 causes of morbidity and mortality. And then as more cancer patients survive due to better chemotherapy and immunotherapy, more patients are ending up with heart failure symptoms due to potential cardiac toxicity from these drugs. And so we're interested in iPS cells to implement the precision medicine concept. In a study done by a former postdoc, Paul Perwich, who's now a faculty in Northwestern, we recruited 8 breast cancer patients, and a year later, some of them have normal cardiac function while others have doxorubicin-induced cardiomyopathy. Traditionally, doxorubicin is the mainstay treatment for both metastatic and early disease breast cancer, but the use is limited because of the intrinsic cardiac toxicity. Now here we generated the patient-specific cardiomyocytes and exposed them to doxorubicin in vitro. We then studied cellular responses by contractility, calcium cycling, metabolism, gene expression, apoptosis, chemoprotection assays. And then we were able to show that the iPS cell-derived cardiomyocytes can be used to recapitulate which patients are more susceptible to doxorubicin-induced cardiac toxicity, as shown in this cartoon by an editorial by Dr. Ting Kemp here. Now, in a follow-up study performed by a former postdoc, Tomoya Kitani, who's now a faculty in Kyoto, Japan, he was able to show that the iPS cells can be used to also model Herceptin-induced cardiac dysfunction. Here we recruited 2 breast cancer patients who did not exhibit cardiac dysfunction, 2 breast cancer patients who had moderate toxicity, and 3 patients who developed severe toxicity from the Herceptin with more than 20% drop in EF. In contrast to doxorubicin, which causes sarcomere disarray and cell death, Herceptin actually is associated with more metabolic derangement and mitochondrial dysfunction. And then importantly, as we typically do, we do a drug screening, and we're able to show that metabolic modulation with AMP kinase activators, such as metformin, can be used to rescue the phenotype. Now, besides doxorubicin and Herceptin, other popular chemotherapy drugs include tyrosine kinase inhibitors, and then common TKIs that you may have heard of include Gleevec and Sutent, which inhibit VEGFR2 and PDGFRB and can cause heart failure as a side effect. And in this study, we generated iPS cells from 13 patients, treated them with 21 most common TKIs, and came out with a cardiac safety index that allows us to predict which TKI is cardiotoxic and which TKI is not. Now, our goal ultimately is to further optimize this cardiac safety index so that we can better predict the safety profiles of chemotherapy drugs before giving them to patients. Now, for the last part of my talk, I want to touch upon how we could use human iPS cells to implement the concept of clinical trial in a dish. Here, the concept is that we should be able to recruit patients, generate their cardiomyocytes or whichever cell type of interest, test which patients respond to drugs and which patients do not, using the iPS cell derivative of the surrogate, and then go back to perform the actual clinical trial on the patients who are positive responders to the drugs. That way, hopefully, you have a higher chance of a success rate for your phase 1-2 clinical trial. So this is an important issue because I think many of you know, on average, it takes about 12 to 15 years and $2 billion to develop a new drug. Even worse, many of the drugs that you take actually do not work at all. And shown here are the top 10 highest sales for drugs in the US. Blue means that the drug works and red means the drug does not work. So for Abilify, which is used to treat schizophrenia, out of 5 people, the drug only helps 1 person. For Nexium, which is used to treat heartburn, out of 25 people, the drug only helps 1 person. So overall, this is pretty lousy response. And this is why the editorial here calls for a time for a 1-person clinical trial. Now, several companies are thinking along the same line. One of them is Takeda in Japan, which has taken advantage of their close partnership with Shinya Yamanaka. This is in April 2015, press release showing Takeda's investment of $268 million into the iPSL technology for developing drugs in the areas of heart failure, diabetes, neuropsychiatric diseases, and cancer immunotherapy. Now, what's amazing is that by August 2017, team has already developed a drug hit based on data on iPSL screening phase 1 and started a phase 1 trial. Here, the investigators generated iPSLs from FOP patients. FOP stands for fibrodysplasia ossificans progressiva, also known as the Munch-Myers disease. It is a rare genetic disorder that occurs in 1 per million with median life expectancy of 40 years. Patients die when their muscle tissues are gradually replaced by bone. And then here, by using a compound library screening of 6,800 drugs, they identify rapamycin as an effective molecule and have now initiated a phase 1 trial. Now, similarly, in the US, companies and federal agencies are thinking along the same line. This is a study published by several companies and FDA using 1 consensus protocol, 2 IPS lines, 3 continents, 28 blinded drugs, 2 statistical models, 10 volunteer labs. And importantly, the investigators were able to show an 87% prediction rate, which is very good considering this is one of the earliest studies by the FDA SHIPA group. And so therefore, there's a significant room for improvement in the future. Now, in our lab, we have also been pushing along that direction. This is a paper we published in 2016 using transcriptomic profiling of iPSL cardiomyocyte to predict individual drug safety and efficacy responses in vitro. Here, we tested 2 drugs, rosiglitazone and tacrolimus. We also use CRISPR genome editing to confirm the potential mechanisms of action as well as adverse side effects, especially for rosiglitazone, which I think, as you know, is a PPAR agonist and was used for diabetic patients in 2000, but was pulled off the market in 2010 because of the increased cardiovascular deaths. And more recently, Naziza Sayed, an instructor in our lab, was able to perform a follow-up study on the large family cohort with Lamin cardiomyopathy, but this time focusing on vasculopathy because we noticed that many of these Lamin mutation patients develop early onset hypertension. In this case here, we differentiated the cells to iPSL-derived endothelial cells, perform a whole battery of assays and functional tests, and then using RNA-seq and ATAC-seq, we identified KRF2 as the potential transcriptional factor responsible for the endothelial dysfunction. And using chemical library screening, we found that lovastatin, which is a potent KRF2 agonist, can rescue the vascular dysfunction. And then the same Lamin patients whom we tested their drugs would then return to our clinic and treated with that lovastatin 40 milligrams daily. The clinical data is shown here. We use EndoPad, which is a machine that can assess flow-mediated dilatation. Normal index is 1.67 and above, and the abnormal is below that. And as you can see here, Lamin patient number 2 started with a poor index of 0.48, improved to 1.06 after 6 months of lovastatin treatment. And Lamin patient number 3 started with 0.61, which improved to 0.95, and then 1.5 after 18 months of treatment. So this is an example at bedside where a clinical observation was made to bench where mechanistic studies were done, and then back to bedside again where we tested lovastatin. Finally, one of the goals in our lab is to start creating a human encyclopedia transcriptomic signatures of drug responses using these cardiomyocytes. I think previously I've shown you Herceptin, TKI, and doxorubicin. And then for cardiovascular drugs, we're using similar model to study calcium channel blocker inhibitor, calcium channel blockers on the left, And then statins, for example, atorvastatin, lovastatin, simvastatin, fluvastatin. And so what we're interested is to understand the cellular molecular effects, which we believe may be independent from the clinical action of these drugs that are used in the clinic. And then I just want to take this opportunity again to highlight the Stanford Biobank, and with the support of the R24, we generated more than 1,000 lines from different cardiovascular history, ethnicity, and sex. And we're basically in the process of finishing up DNA sequencing on all these lines and link it to the PharmGK database so we understand human genetic variation of a drug response. And then we're working with the NIH on biobanking and FDA and drug safety testing. And if you're interested in these cells, so you're welcome to contact me. And then this concludes my talk. I just want to acknowledge my postdocs, graduate students, cardiology fellows, instructors, and former trainees, and also my collaborators as listed here, my funding support. Thank you very much for your attention. I'll be happy to take any questions.